All right, everybody, we're back in the house again with a little bit about the nervous system. We're going to look at three different models today. We're going to look at the model of a neuron. We're going to look at the model of a spinal cord section, and then we're going to look at another model of the spinal cord and its spinal nerves just for a moment. So what we're looking at right now is we're looking at a model of a neuron. And if you're looking at this and you're like, what in the world is that? That really doesn't look much like a neuron. Part of it is because it's got so much detail to it. Another issue is the fact that it's missing some things. For example, if you look at the dendrites, you'll notice that the dendrites here are actually lobbed off. So if I use this laser pointer, hopefully he'll work for me. Yeah, if you see there, you'll notice that the dendrites here are actually lobbed off. They're very short. These dendrites, of course, we know that dendrites receive information for the neuron. So if this happened to be a neuron that was using synapses that happen to be chemical synapses, then we would also see that the dendrites would be the guys that would have receptor sites for the neurotransmitters that were being released from our presynaptic cell. And this cell that we'd be looking at right now, this particular neuron would happen to be a postsynaptic cell. If you look very closely, you can see the little plunger looking structures like this little guy right here. He's representing a synaptic knob or a synaptic bulb, which is at the end of the telodendria. And if we go over here, we can actually see one of the synaptic knobs that are actually cut open or dissected open for us to view. And if you look very closely, you can see the small little vesicles that are inside that are releasing acetylcholine or whatever neurotransmitter it's releasing onto the surface of this particular postsynaptic cell. Depending on the cell and depending on the synapse, that's going to kind of determine what type of neurotransmitter is being released. Now, in this particular model, he's a newer one, so it's kind of difficult to compare to the pictures that you commonly see in your image books. However, the point of this particular neuron model is to get you to understand that Nissl bodies are actually fragments of endoplasmic reticulum. So what you see on this particular model is a lot of endoplasmic reticulum. As a matter of fact, if you zoom in really close, you can see all the crusty, bumpy structures on the endoplasmic reticulum, signifying that those little red flakes are not in fact residue from my bacon bits from my snack that I ate before I did this. However, instead they're ribosomes that makes this rough endoplasmic reticulum. So little pieces of endoplasmic reticulum that would be floating around in the cytoplasm would actually become the Nissl bodies, which is what gives gray matter its grayish appearance. Now, as we continue on, we see there's the little axon terminals, which are all the little rubber plunger things. And then we get to a structure that's kind of awkward. It connects the cell body itself to the actual axon. And that is when we see this right here. This area is known as the Axon Hillock. Now, remember, I said this area. So there's a place here that connects this axon to the rest of the cell body, and that would be the Axon Hillock. What they've done here is they've cut the surface of the Axon Hillock away so that you can see these little reddish fibers here, and these particular fibers are what we know as neurofibrils. So the neurofibrils are inside of the Axon Hillock, and if we continue to follow this on down, we begin to see that this is the actual axon. And what they've done is they've cut away the axon so that you can see the inside of it. Now, there's a lot of things going on over here, so let's just slow down for a moment. Number one, yeah, this is the axon, and this is the inside of the axon. And what you see here with these strange little purple things happen to be mitochondria. However, there is a lot of other stuff on the outside of the axon that we want to pay close attention to. If we zoom out just for a moment, you'll notice that there's something strange going on with this jelly donut looking structure up here, and this happens to be the cell nucleus of an entirely another cell. There's a cell that wraps around the axons of your neurons, especially and specifically the ones in the PNS, and those particular cells are known as Schwann cells, and Schwann cells produce a myelin sheath. Now, Schwann cells are a little awkward because if you looked at a Schwann cell, you would find that it looks a lot like a pancake with this gel-filled middle here, which would happen to be its cell, nucleus. So the nucleus is a part of the Schwann cell, and what it does is it wraps itself around the axon, and it begins to produce layers of lipid that are going to act as an insulator to the axon. These layers of lipid are made out of a lipid substance known as myelin, and when we wrap the myelin around the axon, it creates a myelin sheath. So this dude is kind of like a pig in a blanket. I don't know why I'm talking about food like this, but hey, I guess I must be hungry. So these are layers of the myelin sheaths made out of myelin, created by the Schwann cell, and the outer cell wall or cell membrane of this cell happens to be known as a neurolema. Just like when we studied the muscle cells and we saw that the sarcolemma was the cell membrane of the muscle cell, this is the neurolema of the Schwann cells out here. Also, we'll notice here where you've got one Schwann cell and you have a second Schwann cell, but they're connected at a joint just here known as the node of Ranvier. And if you remember, the node of Ranvier is a section that's very important when dealing with action potential transmission. We don't have time to go into all of that. That's another day and another video. If we continue on down, we also see what's known as the endoneurium, which is wrapped around the individual axons and connecting them, which we'll talk about at another day. And that kind of wraps us up for the neuron model. Now we move on to the next model.